So, good evening, everyone. Welcome to another session of Basic OmicsLogic Training 1 program. Today is our fourth session, and this session, which is still a part of the introductory stage, will be dedicated to learning about genomics and its use in bioinformatics. Today, Mr. Sonny Hamza, our mentor and facilitator, and also our business development director, will be talking about introduction to genomics, DNA variants, and mutations. We will be exposed to genomic data and the use of next generation sequencing to study and understand the information contained within a genome. We will also learn about shotgun sequencing reads, mutations, the need for structured data, and associated bioinformatics methods. We will understand about processing of data for germline and somatic mutations, the analysis that will include mapping, detection of variants, and we also understand the interpretation of significant and insignificant mutations. Mr. Sonny, I'll be handing over to you. Hello, Mr. Sonny.
Yeah, good afternoon, Dr. Adetayo. I will be sharing my slide in the next few seconds so that we can be able to do the needful accordingly. Just give me some seconds. Yeah, can you see my slides now?
Yes, we can.
Yeah, good afternoon, ladies and gentlemen. My name is Hamza Alkali Sani, and today I'll be your facilitator to discussing this germane topic on introduction to genomics. I would like to give a brief introduction of myself since this is the first time I'm going to be interacting with the class. I'm a skilled medical laboratory scientist and also a safety advocate. As a med lab scientist, my interests cut across public health, research, data science, artificial intelligence, and machine learning. My specialty is medical parasitology. I have over a decade experience on that. Another aspect of laboratory medicine, being a laboratory scientist, such as biochemistry, immunochemistry, hematology, serology, microbiology, and clinical pathology. Haven't had full hands-on on the wet lab. The interest to also do research in the dry lab has poured me into data analysis and data science. So today, I am going to be discussing with you, and I'm coming in as a capacity as a mentor in Pine Biotech. I'm part of the OmicsLogic Nigerian team. I'm presently with them as a business development director. And today, we will be looking at a very germane topic, just like Dr. Adetayo has said. And we are going to be looking at introduction to genomics, DNA variant, and mutation. For us to understand this clearly, the reality that comes to our mind is that when we listen about genomics, the first thing that comes to our mind will be genes or code. So this code or gene passed from cell to cell. There are about 3.2 billion letters in total, encoded about 22,000 proteins. And for us to understand this better, we need to get to know that genes are made of biochemical entity called DNA, which in short form is called the deoxyribonucleic acid. The DNA molecule is a double helix that is long, thin-stranded, twisted, and has a spiral-like staircase. In the DNA, the backbone of each strand is made up of sugar, phosphates, and it is also expected for us to note that there are other nucleotide bases here. It is key for us to note that as far as researchers are concerned, in humans, the genes vary in size from 100 DNA bases to more than 2 million bases. An international research effort called the Human Genome Project, which worked on to determine the sequence of human genome, identified the gene that contained, estimated that humans have over 20,000 to 25,000 genes. If you look at the human gene list, there are certain things that is key for us to benefit from there. Over time, it is hard to overestimate the importance of human gene list. I must say that thousands of studies rely upon it, including the effort that discovered how we have complex cases like cancer, certain disorders like schizophrenia and dementia, and Mendelian disorders, and many more. Upon receiving the DNA sequencing result for a sick person or a sick patient, the first question that comes to your mind is, what genes are affected? This very question itself assumes that we know where the genes are. And yet, despite tremendous efforts for the past 2 decades to ensure that we understand how these genes work, it is still a source of mystery for us to unravel. The primary goal of Human Genomic Project, which lasted from 1990 to 2003, was to determine both the DNA sequence and the location of the estimated over 100,000 genes. Scientists at that time believed that once you had sequence in hand, you would fairly quickly be able to determine where all the genes were. Subsequent history have proven otherwise, because the reality that comes to stay is that there are several competing genes database with many thousands of difference among them. And although the number of protein coding genes are gradually converged together, the number of other gene types has exploded. Now, this makes us to understand the types of nucleotide base as it exists. For us to understand better, we need to know that DNA is made up of nucleotides. Now, let us understand about the DNA and nucleotide in a more detailed manner. A nucleotide is made up of 3 components. That is a sugar called deoxyribose, a nitrogen base, and likewise a phosphate group. For the purpose of this discourse, I would just want us to go along, because I know that we have some undergraduates in the team and they need to understand where we are coming from. This is the basics by which research is done, and we need us to make efforts to be able to understand how this works. So these nucleotides are joined together by a chain, by a covalent bond that is between the sugars of one nucleotide and a phosphate and the next, resulting to alternative sugar phosphate backbone. It is important for us to know that nucleotides of 2 complementary strands connected together are called base pairs. These base pairs connect to 2 strands of DNA. When referring to a number of base pairs contained in DNA, we are referring to connected bases. Now, each strand of DNA molecule consists of sequence bases that is complementary to the base opposing strand. What do I mean by this? Pairing between complementary base gives rise to a characteristic double helix shape. Nucleotide base are of 4 types. As you can see on the screen, we have the A, G, C, T. And in this structure, the A is bounded to T, while the G is bounded to C. It is important for us to understand the central dogma of molecular biology. This central dogma allows us to get the good grasp of what we are discussing, to understand what is expected of us. Now, it describes the 2-step processes, which is transcription and translation, by which the information of gene flows into protein, from DNA to RNA to protein, as described on the diagram. You know, one of the things that we have to get clear in research is that how do we bring all what we have said together into the next generation sequencing? Having understood this, it is key for us to note that with the advancement in NGS technology, that is the next generation sequencing, that there is an ever-increasing number of genome sequence, and the cost of sequencing is drastically reducing, and as clearly indicated in this plot that you can see here, following the Moore's law. If you can look at very much well, in the year 2002, the cost for you to do a genome sequence is about 100 million. Whereas in 2017, it will just cost you $1,000 only. And this goes to tell us that as the year progresses, there will be a very significant increase in our technology. We should be able to sequence more genes as possible. According to Moore's law, it states that we expect that the speed and capacity of our computers to increase a couple of years. We will pay less for genomic sequencing. The first genome sequence was done in 2007. The first tumor that was sequenced was done in 2009. Today, we have over 20,000 tumors that have been sequenced for far less the amount. When it cost like $1 million, $100 million in 2000, today with less than $1,000, you can get a complete gene sequence. This goes to tell us how research has gone to benefit mankind, particularly when it comes to genomic sequencing. It is key for us to understand that NGs allow researchers to explore genetic variation like never before, down to a single nucleotide resolution. With the next generation sequencing that we are talking about today, whether you need to identify specific variants from a focus set or a region, or look more broadly for potential causative variation, the whole exome sequencing or a whole genome sequencing both offer effective solution. Whole exome sequencing is targeted to view the protein coding region of the genome. It offers a reliable and sensitive detection of coding variants. It is fast and cost-effective. It is key for us to understand that whole genome sequencing, on the other hand, provides a comprehensive view of the genome as far as coding and non-coding and mitochondrial DNA is concerned. It is a reliable and sensitive as well, and includes detection of all variants type. It's low cost and fast method for library preparation. Distinguished participants, I'd like us to understand the types of the NG sequencing that we have existing. So I want us to understand this sequencing technique. First and foremost, we'll talk about what is known as targeted sequencing. Now, targeted sequencing, here we can investigate these specific regions from our sample and completing targeted sequencing. Targeted sequencing provides us both high coverage and in-depth look at what happens within specific region. Such localized sequencing might be useful if wanted to identify mutation that you suspect lead to a particular disease, such as cancer. Targeted sequencing works best between 40 and 400 genes. It is ideal for looking closely at small, specific region of genome. It provides high coverage of gene and allows us to see single base variation, such as insertion and deletion. It also generates a small amount of data, potentially allowing for easy analysis than some of the larger scale sequencing techniques. Then we'll talk about whole exome sequencing. That's the second one we want to look at. In order for us to examine genomic DNA on a larger scale, we can run whole exome sequencing, which focuses on the protein codings. Now, this gives us a broad view of genome sequence. The major advantage here is that whole genome sequencing allows us to see both coding genes and the non-coding region, providing us more information about the information within the genome. So now let us understand what the different types of sequence reads based on length, size of sequence. We need to understand that small reads sequencing up to 250 to 300 base pair up till now, but the 100 to 150 base pair is still the base standard. Packbell and mini ion, about greater than 50 kilobytes, but with a very large range of reads length in the same run. Then we'll talk about the short reads as significant and sequencing application known as genome reference. Longer reads are beneficial for de novo genome assemblies and structural variation detection. Over the years, advances in NGS technology provide us with different types of sequencers for each types of reads. We can see what we have for short reads and what we have for long reads. Now we take time to discuss the types of mutation that exist. Usually, DNA is an incredibly stable molecule that allows our genetic codes to be maintained and ultimately passed onto offsprings. Despite the stability, there are some instances when sequencing within the DNA changes. Now, these changes are referred to as mutation and may be detrimental effects on the DNA code. The types of mutation is characterized by changes it produces within the DNA sequence. And what we discussed now will be called the point mutation. Now, point mutation results when there is alteration at a single base or a nucleotide. Now, these three types of mutation we should become familiar with are, as we have on the slide, substitution, insertion, and deletion. In addition to point mutation that affect single base in the DNA, there are also mutations that change more than a single base. This mutation can affect a whole section of the DNA, dramatically altering gene expression and protein function. For example, a DNA sequence that is inserted improperly or more times than normal results in an insertion mutation. An inversion results when a sequence of base reverses orientation within a DNA molecule. Another type of mutation is known as translocation. Translocation can happen in two different ways: within the same chromosome or between two chromosomes. It is key for us to understand that with what we have on ground, we need to look at mutation that can be encountered and the impact they have on genome. Now, they are of three kinds. The first we're going to discuss is the missense mutation, a genetic alteration in which a single base pair substitution alters the genetic code in a way that produces an amino acid that is different from the usual amino acid at that position. Some missense variation or mutation will alter the function of protein, and this is called the missense variant. The second one we need to look at is a nonsense mutation. Now, this also involves a single alteration to the DNA base pair. However, in the case of nonsense mutation, a single change in the production of a stop codon, thereby terminating the protein synthesis prematurely. As a result, there is an incomplete protein formation formed that may function or may not function. And this is known as the nonsense mutation. And finally, we have the frame shift mutation, which occurs when any addition or deletion mutation results in a change of the gene's reading frame, which includes a group of 3 bases that encode for amino acids. The change in the reading frame alters the grouping of the base and subsequently changes in amino acids that are encoded. As a result, the encoded protein becomes non-functional. This will now lead us to discuss codons and amino acids, it is key for us to understand that a change, even at a single base level, can be enough to change the type of protein expressed. So let us see how these happen and appreciate the variation that exists. Each amino acid is represented in the DNA as a set of 3 bases, referred to as a codon. For example, the ATC, for example, a mutation in a codon sequence can result to a change to the amino acid produced. Now, it is key for us to note that results in altering the structure and the function of protein. If you look at this table here, you can see how changes in codon could impact the amino acid expressed. Thus, both insertion and deletion can change the DNA sequence at the RNA and the protein level by changing a particular codon as expressed. So at this junction, I want to pause if you have questions for us to look at. Do you have questions? If you have your questions, can you post it on the chat box for us to look at? Then we can proceed. Any questions? So while no question is ongoing now, let's move on. Questions?
Mr. Sonny?
Yes.
There are 2 questions presently.
Okay.
Is asking that the table showing the positions. I don't know what he means by that. Maybe you can expatiate on that.
The table showing.
Mr. Ayomiko?
Yes, sir.
Can you expatiate?
Yes, I just had to put something because I felt you wanted to move on. The table here that is showing on the screen, yes. I could see the 4 nucleotides. That's the T, the A, the G. And I can see that you have them in 3 different parts.
Okay.
Yes, so horizontal, vertically, and the other one at the other side. Then if you now look at each cell, you have 3 of those things there. How does all this come together with the DNA and the characteristics that they kind of give?
Okay. Yeah, thank you very much. Now, if you listened very much closely to what we said earlier on, we're talking about mutation here. Now, any alteration that results to the structure and the function of protein here completely have a change in the codon. And this inevitably affect the impact in the amino acid being expressed. Now, the alteration itself has effects, and as such, what we get at the end of the day gives us a different expression. That is what we're trying to explain clearly here. Now, if you look at our previous slide before this, where we talked about the types of mutation, we talked about the mean sense, the nonsense, and the frame shift mutation. We clearly explain that all this happened as a fallout for genetic alteration of a base pair, either by substitution or any kind of alteration. And when this happens, it has adverse effects on what is expressed at the end of the day. And as such, the variation gives it rise to changes. Now, if we are going to go further to the next slide, we'll look at the significant effects of DNA variation when it comes to diseases. Just like I said earlier on, when they did the genome sequencing, the actual thought was that since the whole human body genome has been sequenced, we already know what it is. The question is, if you have a sick patient that has cancer or any form of disease, and with whole body genome sequencing, we should be able to specifically say, this is where from the gene sequence, this is where the cancer is, and this is what we need to do to alter and be able to sort the challenge. But unfortunately, it is not like that. So the moment an alteration happens, what comes out after it varies. So as such, the concept here is for us to understand that this alteration comes out with a different disease condition. And as researchers, what we're now trying to do is how to be able to identify where the disease is. So if it were to be very simple, the moment we have our human genomic sequencing done, we should not have a challenge with cancer for now. But today, cancer is still a problem. We should always say, with this gene identified, this is where the challenge is, this is where it is. But because of the variation, we still need to try to research and try to see how far we can get the outcome. So every variation itself gives us a different outlook, which will make it difficult for us to pinpoint. Let me further express this by the next slide. Probably this will give you a better view to it. Like so far, we have tried to understand the concept of types of mutation. Now let us understand why and how mutation plays a significant role, like for example, in cancer development, and or better still, how we derive for tumor formation. This will give us a better view on what we're talking about.
Sonny,
yes.
Mr. Sonny, before you continue, there are still other questions on the chat box.
Okay.
So
what are questions?
Yes, Promise Nwafo said, can you please explain missense and nonsense mutation again? While Izudin asks that, do deletions happen only in point mutations?
Okay. Let me take you back. After going back to the slide for missense mutation. Missense mutation clearly states that when we have a genetic alteration in which a single base pair substitution alters the genetic code in a way that produces amino acid that is different from the usual amino acid at that position. Now, I don't know if that itself produces what we call a missense variation. This is when we have a single base pair substitution that alters the genetic code. While the difference with that, while for nonsense, this involves a single alteration to the DNA base pair. However, in the case of nonsense mutation, this is a single change. You get what I'm saying? For nonsense mutation is a single change. While for missense, missense mutation, what we have is a single base pair substitution that alters the genetic code itself. What's the second question you asked, Dr. Aditayo?
Deletions happen only in point mutations? Is it only in point mutations that you have deletions?
No. Let me go back to the slide for that so that you can be able to look at. Just like the question is deletion, I didn't get that again.
That deletion, when you have a deletion, does it only happen in point mutations? Are there other forms of mutations that deletion can lead to?
Okay, basically it happens in point mutation. Deletion happens in point mutation, basically. It is most expressed in point mutation. And the reason being that if you had gone through introduction to genomics in the on the platform, it clearly states there that when we talk about point mutation, just like I said earlier on, we have issue of substitution, insertion, and deletion. And this is a function that comes based on the changes that occur. And these changes that occur completely change what we have in the codon. Now for substitution, just like I can see on the slide, you have a nucleotide being substituted here. While in insertion, we have it being replaced. For deletion, a complete removal itself gives us completely a different outcome in the amino acid. Now, this variation itself gives rise to something entirely new that is an aberration that we need to look at while doing other things in the research work. I don't know if that is. Do you have any other questions?
For now, there are no other questions.
Okay,
so to make it clear,
I don't know. Can I come in, please? Just
okay.
to add. All right, thank you very much. I'd like you to go to the slide showing the codon table, please. The question that was asked by the first person who asked a question. Yes, this particular slide. From what we understand, amino acids, right from the central dogma of molecular biology, we know that the amino acids are formed from, the set of 3 nucleotide letters or nucleotide, that forms what we call the codons, like we were told. So each of these letters, by the time the sequence is being read, interpreted to, amino acids respectively. And so this table on to the right actually explains, how these sequence are being interpreted, how these codons, form the amino acids that go on to eventually give us a protein that will be expressed subsequently. So, like on that table, you could see TTT. So, if the nucleotides are being read and you see a set of 3-letter T's, okay, that is thymine following each other, the chances is that they will be, when during translation, they will be forming phenylalanine amino acids, okay? All right, so, and then it will go on like that. We have others. So each of the things we see in that table, is also telling us the set of 3 nucleotide codons, the set of 3 letters that, and what they interpret to mean. And this is where it actually comes in what we are doing here, talking about mutation. So you could imagine if the letter were to follow the sequence of, say, TTT, okay, the first one on that, in that box.
As shown here.
Yes, which is to interpret, to form the amino acid phenyl, and so to maybe to point mutation or what have you, changes to T, let's say TTG. The last letter T there is suddenly changed either by deletion. Of course, if there's a deletion, the next set of letters that will, I'm sure G will have started the next set of 3 letters, but now it's the last letter forming the set of 3 nucleotide that needs to form this particular amino acid. So, of course, it will be interpreted and then it will produce the amino acid leucine. Okay, and just to wrap up my contributions here, we know, for example, that for sickle cell patients, the singular difference in their own, hemoglobin that changes the hemoglobin A to S is just the, presence or, mutation around the amino acid valine in their hemoglobin chain. Okay, so, and you can imagine the normal amino acid that should have been there. I can't recall specifically now, but then based on the mutation, the misreading of the set of nucleotides, suddenly we find valine. I'll call that up before we end this session. Okay, we suddenly have valine replacing the amino acid that should have been there, and because of that, the blood cells become sickle, and then the whole lot of suffering that is associated with sickle cell. So that is a typical example or impact of either a deletion, or insertion or frame shift or what have you. So that is just what I want to explain. And to that particular question regarding the codons and these tables that we have here.
Thank you very much, Mr. Adekole. Dr. Adetayo, you have any other contribution?
No, Mr. Abdulaziz also has a question that we haven't answered. And he's saying that why point mutations? Why did you isolate point mutations? Why explain other types of mutations? Are point mutations the only type of mutations we have? Why did you isolate it? Why explaining other points of mutations? That was his question.
Now, for the purpose of this discussion, when we talk about variation, we have to discuss point mutation for us to understand how things happen, particularly that to give us, just like what Mr. Adekole has said now, differences, sorry, let me go back to the slide. Now, particularly on how Mr. Adekole has explained clearly, the essence we have to pinpoint point mutation is to be able to express and explain to us the variation based on the discussion we're having as regards what happens in terms of substitution, insertion, and deletion as a type of mutation that exists within the genomic, at the genomic level. That's the essence, basically. I don't know if that clears the...
So, Mr.
Yes.
Mr. Ayomiko,
are we good to go?
Mr. Ayomiko, have we answered your question?
We have different types of mutation. Let me just quickly add. Of course, we are focusing more on point mutation because that is one of the most common, set of nucleotide changes and mutations that has a lot of impact, especially when we are looking at genomic variants and how this readily happens. You remember the other time, Mr. Sonny mentioned that the DNA is fairly stable. However, during the process of replication and transcription and the rest, and passing all of these genetic information from generation to generation, mutation, fairly happens almost. I mean, it happens almost very frequently. Let me use the word. Let me explain it that way. And in fact, as we are humans, as I am now, of course, I don't have any disease condition I can say is associated to this. But then mutation is happening in us. It's happening. Whether we will not transfer it or to our children or so, I mean, their offspring is another point entirely. And thankfully, we have a lot of DNA repair mechanisms, that helps us to guard, to prevent these mutations from being passed across, these, DNA changes and damages from being passed across. We have those mechanisms in our body, and it's actually happening. So point mutation is one of the most common types of mutations that happen almost fairly all the time. There are chances, of course, he mentioned some other ones. We have the translocation, we have the, he mentioned them, the translocation and rearrangement and all of that, okay? And some of them has to do with deletion of, a whole section of the gene. I mean, quite of translocation from one chromosome to another, or even within the chromosomes. But these are quite rare compared to when just letter A is deleted or letter A is inserted during replication and the rest. So point mutations happen fairly more, more frequently. Even in normal humans, the chances are just that it might not be transferred, down to the offspring and the rest. Okay. It happens. We are exposed to mutagens. We are exposed to several things that can lead to mutations almost every day of our lives.
Yes, okay. Moving forward, we look at, so far we have tried to understand the concept and types of mutation, and we particularly picked on point mutation for us to discuss. Let us now understand why and how mutation plays a significant role, like in cancer development, or better still, how it drives for tumor formation. As we know, there are various types of gene, important genes that play a role in cell and regulation in cell growth. Any disturbance in any of these genes is a dysregulation of cell growth, eventually results to a tumor development. Now let us understand certain things that is key. One, there's tumor suppressor genes. Usually these genes regulate the cell growth, thus they are considered as protective genes. We have what we call the oncogenes. Normally these are genes responsible for growth cells. If any mutation in them results in promoting their normal function, this will result to uncontrolled growth. And thus they are key players that can turn normal cell to become cancerous cell. Now it is key for us to note that when it comes to DNA repairs, here mutation can be of different types. It is key for us to note that we have acquired mutations, and these are most common cause of cancer. And we have the germline mutation. These are the less common cause of it. I would like us to x-ray how the process happens so that we can be able to appreciate the reality when it comes to be working with the pipelines. So there's a transition from pre-analysis from the dry lab to the wet lab to the dry lab, and likewise post-analysis that we need to understand. And also look at the challenges that exist when it comes to diagnosis. Let us understand the approach and challenges of diagnosis based on whole exome or whole genome sequences. Now let's look at the different challenges that in the various steps. Now, first and foremost, we have the pre-analytical phase. This is the initial testing phase. Challenges that comes with this phase affect status may be assigned incorrectly. Sequence variant detected in family members may mistakenly be labeled as unaffected. And this can happen as a challenge in pre-analytical phase. At the analytical phase, probably when you have the wet lab, where we do the gene sequencing itself. One of the things that happens in the wet lab is that, one, we have sort of false negative results. That is, region that cannot be sequenced will not generate the variant for downstream analysis. Then we also look at having passed through the wet lab. Where we come into play most often in data science is the dry lab, that is the bioinformatics method. Here we have about 4 basic concepts that have challenges. As regards the short reads alignment, the challenges that we face there is sorts of false positive results. This what happens if you have short read aligned at incorrect position at a reference. It can be incorrectly identified as mutation. The second challenge we might have is probably in genotyping. And this certainly decreases the low coverage region. Thus, as such, can be discarded. Otherwise, might be important. Also, we have variant filtration. And this error occurs when the filtration assumptions are violated. Then lastly, we talk about post-analytical. As regards post-analytical, challenges that we have involved is that at this step involve judgment of analysis by researcher and clinician. So there's a risk for considerable amount of judgment and exploration, which can happen in case of rare or new variants. So one of the main concepts of genomic sequencing, particularly for data scientists, is for us to be able to reflect very much well and do an analysis clearly when it comes to moving from the pre-analysis from wet lab down to dry lab. And this go a long way to explain to us that as researchers, we need to make efforts to be able to do more of critical thinking than thinking of making efforts to run pipelines. Finally, we'll look at how it happens when it comes to cancer itself. As I said earlier on, we have acquired mutation, germline mutation, and these are clearly stated, as stated earlier on. And having understood these basics, the next phase is, as a data scientist, how do you come on board to be able to do your analysis and do your reads clearly with putting in mind how to avoid those challenges we listed above. So at this junction, I'm going to hand over to Dr. Aditayo Aborishade, who is going to talk more about how we're able to run genomic pipelines, explain to us what we expect to know, and see how far we can move on from having the basic knowledge of genomic sequencing, then how do we move from the wet lab to the dry lab and do analysis without any hitches. So Dr. Aborishade, you have the floor now.
Thank you, Mr. Sonin, for your presentation. I would like to share my screen. Give me some seconds, please. Can we all see my slides? Hello, can we see the slides?
Yes.
Oh, thank you very much. I think the first clarification I would first like to make is on the point of point mutations, because most of the questions in the chat have been regarding this. Now, there are different forms of mutation. Mutation can be small scale, involving few bases, and it can be on a large scale. But the most common form of mutations are the ones that involve a single nucleotide base. And that's why we call it point mutations, or otherwise can also be known as single nucleotide polymorphisms. Meaning, in this case, the variation in the genetic makeup only involves one nucleotide. And there are different types of them. The most common types are this silent mutation, in which even though there is a change in the nucleotide, there is no concurrent change in the amino acid that it translates to. Meaning that, let's assume I'm supposed to get alanine. There is a change in a nucleotide, but the outcome is still alanine. That implies the point mutation does not lead to a change in the amino acid and subsequently the protein produced. In that case, we say this mutation is silent. The second one is what we call missense mutation. In this case, the change in the nucleotide base will equally lead to a change in the amino acid formed and consequently the protein formed, meaning that there is a problem with sensing of this base. It was supposed to produce alanine, and the mutation involving the nucleotide base is now producing something like glycine or leucine. In this case, we call it missense mutation. The third one is what we call nonsense mutation, in the sense that the nucleotide change or variation that happened in this case is going to lead to an amino acid that is known as a stop codon. By formation of a stop codon, there is a termination in the elongation of the amino acids that will lead to that protein to be formed. Meaning that if you're expecting the protein to be 5 amino acids or 10 amino acids, at the end of the 10th amino acid, the next sequence will give rise to a stop codon that will lead to termination of the protein. So now assume that at the second stage, so let's assume that at the second stage, there is formation of a stop codon, which are usually 3 types. There are 3 types of triplet codon that lead to stop codon. 1, TAA, 2, TAG, and 3, TGA. Any variation in the point mutation that will lead to the formation of any of these 3 stop codons will lead to termination in the amino acid elongation for a protein. Therefore, we call it nonsense mutation because it directly leads to termination in the elongation of chains. Now, aside point mutation, there are also other types of mutation. right? One other common one is frame shift mutation. And this is usually caused by insertion of a base or deletion of a base. These are not as common as point mutations. And in other series, in further courses, we might delve into it and talk about it in depth. But for today, our focus is on point mutations, single nucleotide variations, or also called single nucleotide polymorphisms that could lead to either disease condition or increased susceptibility of an individual to a disease condition. So now, once we have done our sequencing, we are probably thinking of, we are trying to compare a normal human tissue with a cancerous human tissue. We have sequenced both. Now we want to do our analysis. How do we check for variation? And how do we assume or presume with our research that maybe is the variation in these nucleotide positions that induced or caused the susceptibility of this client or patient to the cancer or disease condition? This process is what we call analysis. So the first process here is after we have done our sequencing, we need to pre-process the results or the reads that emanated from the NGS. Remember when we are talking about NGS, we mentioned that it is not all reads that have enough quality to give us an high confidence about the calling of that read. So during pre-processing, we want to trim away all the reads that their Fred score is beneath 20%. Because if we don't, by the time we start analysis, we might end up seeing a variation. I might assume it is actually a true variation, not knowing that it is a mistake based on the poor quality score of that read by the NGS. So the first thing we want to do, we want to trim all these poor sequences. Secondly, doing NGS and doing library preparation, PCR is done to be able to form clusters, and thereby doing the sequencing, we might end up having what we call technical duplicates. So part of the processing is we also need to remove these technical PCR duplicates, and also the adapter sequences that are added to the ends of the fragment during NGS. So all these processes that we do before we begin the main analysis is what we call the processing. So when we are done with that, the second stage, the second thing we do is what we call mapping or alignment. As we said, we do have a reference genome that serves as the control for all our sequence reads. So after, before the sequencing, the genetic makeup or the genomic bases are broken down into fragments before they are read in short base pairs of between 100 to 150 kilobase pairs. So to know the positions of what we have sequenced, to know whether our sequence belongs or fits into chromosome 1 or chromosome 2, the positions in their chromosomes, we need to align them and map them to our reference genome. I remember we said that the present build of human genome that we have is version 38. So that's the second thing. And to do mapping or alignment, we have several software that can do that. We have Bowtie, we have TopAt2, we have ISAT. Then the next thing is, after this mapping and this alignment, we then need to call the variant, meaning we then determine positions on our sequence, on our reads, whereby there is actually a change nucleotide from the reference genome. Meaning, after mapping, we'll be able to visualize where exactly on our sequence read differs from the reference genome. Is there a position 102? Is there a position 500? We'll be able to see during visualization that, oh, this is where there is variation. And two, we should also need to know that not all variations lead to a disease state. Not all single nucleotide variations lead to cancer. Not all variations present with a disorder. There are some variations that are perfectly normal in some group of people. So in our variant calling, we need to be able to marry this together so that we don't assume that every variation is a cause of cancer or disease state. That phenomenon of trying to rule out variations that lead to disease states is what we call filtering for significance, where we account for a variety of factors that could make a variant more or less significant. And the factors to think about in this case is coverage, is the overall mutational load in the sample. When this is done, our next step is we need to calculate differences. For example, we are trying to compare a mutated group where patients have cancer and a normal match sample. We will now compare those two reads against the reference genome and measure significance. How significant are these variations in deciding whether this is the cause of this disorder or this is the cause of this problem? And lastly, we'll do what we call annotation. Annotation is after discovering the segment or the positions where there are variations, we need to now take our result to an annotation database that will now tell us the gene or the group of genes that are present in the area where we have noticed or identified mutations or variations. This is the pipeline that we usually run. And this is also available on the Toba side. However, we will run this pipeline today. The running of the pipeline, the mapping, the data mining, the visualization, and the variant calling is the goal and the meat of our next session, which is next week, Tuesday. But this is just a pictorial illustration of how to run this data. Once you have your reads, you can go under the genomics epigenetics tab and click on mutation to variant analysis. From there, this is the normal pipeline. That once you click start, you first want to use to clean the PCR duplicates by making use of what we call PCR clean. This removes PCR duplicates. Once we do that, the next step is to sequence using Bowtie. Bowtie 2 is usually the preferred tool to use in this case. And once that is done, the next thing is to use a variant caller, which is called Striker. It's also on the pipeline. After Striker, you can go straight to end the pipeline, or you can use an adjunctive variant caller known as somatic sniper. When this is clicked, then we can end the pipeline. Once the pipeline is clicked, the next thing is to allow our platform to run its results. Now, once the result is achieved, we can then go straight to our browser or integrated genomic variant viewer to view the variations in our reads. I'm sure for some of us, at least for some of us that have been reading their chat or asking us questions, we know most of you have this IGV already on your system. If not, we'll employ you and join you to please download it. So by the next time we'll see, that will be next week Tuesday, all of us will have it on our system and we can go step by step in learning how to import our sequence reads into the pipeline, how to do pre-processing, how to map, and how to call the variants, and how to analyze whether these variants are actually the cause for the disease condition or the cause for the patient's susceptibility to cancerous lesions. So by next week, we'll be able to talk about this and describe the pipelines in total. At this point, at this junction, I want to ask, are there questions or clarifications or queries that you want me to answer or want any of the mentors to answer? Questions or queries?
Dr. Adetayo, there's a question from Sarah in the chat box.
Okay, I've seen the question. Sarah, you said, at what point do we determine if a gene sequence is novel. I want to assume maybe you mean, at what point do you determine if a mutation of a gene sequence is novel? Now, when a researcher wants to ask a question, the first thing to do is to already determine ab initio. What questions do you want to ask before you sequence? If your goal is to look for mutations in an already defined gene, in a genetic disorder whereby the gene causing this genetic disorder has been identified, then at that point, the type of sequence you do is targeted sequencing. You already know the genes that cause this variation. You are only interested in knowing whether the same variation is seen in the subset of patients. At that point, you target the genes or panel of genes and you sequence them. In cases whereby you don't know the gene implicated in a tumorigenesis, or there has been no robust literature review to identify the genes or panel of genes involved in a mutation, that is the time we do what we call whole exome sequencing. In that case, you sequence all the genes available. You sequence all the protein coding regions available. And that way, by sequencing, you then have the likelihood of being able to detect the actual mutation that could be the cause of the disease condition or the tumor condition at hand. And thirdly, when you, or probably you do not know the genetic mutation causing this disease or the epigenetic causes. Now, aside gene mutations, it has been proven by literature that there are other effects that are not part of our genetic makeup that could either silence or inhibit the expression of some of these genes. In that case, even though there is no variation in the gene makeup, the epigenetic effect could affect the expression or regulation of this gene. If that's what you are interested in, you go ahead and do whole genome sequencing. So by that, before you even start out, you would have made your decision. Am I trying to identify a no gene? If that's the case, do targeted sequencing. If you are trying to identify a novel gene and a gene that has not been identified, your goal then is to do whole exome sequencing. I hope that would answer Sarah's question.
Yes, I think to a large extent it should. And I'll just add a few slides. Can you hear me? Hello? So a typical example would be what happened during the novel coronavirus. Of course, today there is a reference genome for coronavirus, which is the sequence that was produced in Wuhan. Okay, but of course that is why it's called novel coronavirus, because based on alignment, I mean there are certain guidelines as to the amount of variation you would have before you can actually call a particular sequence novel, meaning it's new, it hasn't been in existence before. So there were certain of those guidelines that were met with that particular sequence, and that was what led to that virus being tagged a novel virus. Okay, so essentially alignment and all of the things Dr. Adita had explained, come to play in determining the sequence you have at hand and the interpretation that will be given to it. Okay, Dr. Adita.
Thank you, Mr. Adekunle. So, Sarah, yes. You said I should repeat my last statement. So I'm going to say this again. When you want to identify a gene that has already been characterized and identified with a cancerous lesion or a disease disorder, the sequencing you do is called targeted sequencing. Targeted because you are targeting a known gene. If your goal, however, is to identify a novel gene, a genetic mutation that has not been characterized or identified with a disease disorder or cancer, the type of sequencing you do is called whole exome sequencing. In that way, you sequence all the available genes that are known in the genome, almost 20,000 or thereby. By sequencing the old number of genes, you can then, during your analysis, be able to either identify a genetic variation or a genetic mutation that could have led or that could be the cause of that tumorigenesis. Thirdly, if your goal is not only to know the genetic mutation causing a disease, but also to know corresponding or adjunctive epigenetic effects that could either lead to silencing of a gene or active transmission of a gene, thereby leading to a genetic variation or tumor. In that position, the sequencing you do is all genome sequencing. That way, not only would you be able to sequence the protein coding regions of the genome, you will also be able to sequence the non-protein coding regions of the genome, because these non-protein coding regions have been characterized and have been identified to have effects such as silencing a gene or increasing the capacity or ability of a gene, even though they are not in genetic makeup, but their regulatory effect could lead to mutation or other effect that can lead to tumorigenesis. So if that's your goal, then just do whole genome sequencing. I hope I've been able to answer that. We are pressed for time, so if there are no questions, we would like to say that the next session, which is on Tuesday, will focus basically on running or doing the genomic analysis that I've outlined. We'll talk about how to search or access data sets to get these genetic sequences. We'll set up a relevant pipeline where we'll do preprocessing, where we'll map to reference genome, where we'll visualize a sequence, and then where we'll end up calling for variants or variations in this sequence, and where we can then search for significance on whether this disease disorder or this cancerous tissue or cancerous condition is caused by the identified genetic mutations in our genomic sequences. So our assignment for next week, we have noticed that some people have still not completed their profile on the Edu Biofile platform. We beseech and implore that for the purpose of community building and community interactions, we employ them to please complete their profile. For people that have still not completed Genomics 1, this week or this weekend will be the best time to complete Genomics 1. So by Tuesday, when we start discussing the pipeline, it will be easier to understand and to follow along. And we also ask that for the people that have done Genomics 1, please keep running the demo pipelines for visualization and mutation analysis. Don't run a pipeline once and assume or presume that you have availed yourself of all the knowledge on that pipeline. You can run for as many times as possible. Begin to manipulate the pipeline, begin to understand the pipeline. So by the time we come on Tuesday, it is easier to follow and understand. Again, I implore that we should all please download IGV on our system, because it needs to be resident on the laptop. It's free. The website is shown on where you can get the links, are available on Genomics 1 course. Please avail yourself of that. Then also, if you've done all this and you still have some time on your hand, you can as well go ahead and complete the coding lessons. The one specifically for Genomics 1 is how to read file and sequences, how to read DNA sequences or protein sequences into how to Python. Lastly, we would like to call on the program director to say a few words before we eventually close the session. But please, in case you have questions, you can still use the form on our organization page. You can use our WhatsApp chat group, and you have all our individual numbers. You can call any of the mentors for clarifications, more answering of questions, and the likes. Thank you very much.
All right, thank you, Dr. Adetayo. Yes, thank you very much, Dr. Adetayo. We want to thank all our participants for joining today, and please let's take all of those assignments but particularly concerning the first task, which is completing the profile. Please, this is very important. I would like to state at this point that if you have not already done so, I mean, you stand a chance of likely not having your certificates, aptly written. Take, for example, when you registered, you have registered with Toastman or some nicknames or your email address, and that is what you have there. So it is when you complete the profile, you go on to change the name to add your real name, Adekunle Adeboye. It is at this point, because that is what will be used automatically. I mean, it is not done physically. The certificates are not written by hand. It is computer generated. So it is the name on the profile that will be automatically used to generate the certificate. And we can see that already for those of us who have been completing some of those asynchronous courses. So please, for those who are here to complete the profile, do well to do that and add, go ahead to populate all the forms and all the feeds in the profile sessions that needs to be completed. And then I like to add quickly before we end, please, we don't want a situation where we participants already ask a question about some of the free courses, the asynchronous courses, transcript tools, and the rest, and then we get maybe is were intimidated or not. No, we want to carry everybody along. That is why we still kept to the the scheduled outline to ensure we take some of these things. I'm sure some persons who have taken the journalism who have completed it already will be more familiar with some of already done. So please, because our goal is to ensure we carry everybody along and everybody gets something out of this training. So please do not be intimidated. Please get like 6, 7, asynchronous courses certificate, while some get 3. What matter is not the number of courses you complete, but ensuring that we carry you along and we're able to get the basis of what the OmicsLogic and bioinformatics tools that are available to be able to move on, with these basic platform. I'm sure that when we move to basic part 2 and the rest, some of them begin to form a bigger picture that we all desire. So I want to appreciate every one of us. If there are no other comments, and like Dr. Aditya said, please, we can keep it on our forums page as well as on the WhatsApp group. But at this point in time, I think it would be safe to call it Uhuru and end this section. So I want to appreciate also Dr. Aditya. Over to you, please.
Well, without further ado, if there are no questions, I think on our WhatsApp page for the next session on Tuesday. Thank you, everybody, for coming today. I wish you a very nice and lovely weekend. Bye-bye.